Hi students, myself Sadesh Kumar Konidala, Assistant Professor, Department of Pharmaceutical Sciences, Vignan's Foundation for Science, Technology and Research, Guntur. In this video, we are going to discuss about the metabolism of nucleotides. So we know that nucleotides are the nothing but these are the compounds which are useful for generating the genetic material, that is nothing but the nucleic acids like RNA and DNA. So the DNA and RNA is the composition of nucleotides. So the nucleotides are the basic molecules that are useful for the synthesis of RNA and DNA, that are nothing but the genetic materials. Okay, so these genetic materials are useful for transfer the characteristics or properties of the parent to the newborn babies or further generations. Okay, so let us see how the nucleotides are subjected for the metabolism. So we know that these nucleotides are composed of by nitrogen bases, pentose sugar, phosphate. So as I already said that nucleotides are, so these are the basic material for producing of DNA and RNA, and these nucleotides are composed by the nitrogen bases, nitrogen base, pentose sugar and phosphate. So by combining these three, we will produce the nucleotide, and this nucleotide combine to form the RNA and DNA and RNA. Okay, so first we'll see, so what are the nitrogen bases or pentose sugar, phosphate present in the nucleotides. Okay, so before going to discuss all these things, we should know what is a role of these nucleotides in the biological system. So these nucleotides are useful for building the blocks, or it will act as monomeric units in the nucleic acid. So that means I already said that nucleotide is nothing but the, so this is the basic material, or it is a building block of DNA and RNA. So that is the genetic materials. Okay, so these are the monomeric units of the nucleic acids. Nucleic acids are nothing but the DNA and RNA, and they also act as the structural components of some coenzymes of B complex vitamins like FAD, NAD+. So these coenzymes, these are the coenzymes FAD and NAD+, these are the coenzymes of B complex vitamins, coenzymes of B complex vitamins. So these nucleotides will act as the structural components of coenzymes of the B complex vitamins, and also used in the energy reactions of these cells, like ATP is in the form of energy currency, or so these type of reactions also these nucleotides are utilized, and also used in the control of metabolic reactions. So these nucleotides are also used in the, or also plays the major role in control of metabolic reactions. So these are the roles or functions of the nucleotides in biological system. First one is they will act as the building blocks or monomeric units of DNA and RNA, that is nothing but the nucleic acids, and they also act as the structural components of some coenzymes like FAD, NAD+ of the B complex vitamins, and also plays the major role in energy reactions of the cells like production of the ATP, and also plays a major role in control of metabolic reactions. So these are the some functions of nucleotides in biological reaction. Next, coming to the, what are the nitrogen bases which are present in the nucleotides? So the nitrogen bases which are present in the nucleotides are nothing but purines and pyrimidines. Purines and pyrimidines. So here we can see the structures of purine and pyrimidine. So here so the structure of purine is nothing but consisting of 4 nitrogen atoms in the heterocyclic rings, This is the purine ring. And coming to the pyrimidine ring, so this is the pyrimidine ring. It consisting of 2 nitrogens in their aromatic ring. Okay, this is the pyrimidine ring. So the nitrogen bases which are present in the nucleotides are nothing but, so they consisting of a aromatic heterocyclic compound. In that aromatic heterocyclic compound, in case of purine, consisting of 4 nitrogens, and in case of pyrimidine, consisting of the 2 nitrogen. Okay, so the bases are of 2 types, like purine and pyrimidine. Purines are numbered in the anti-clockwise, pyrimidines are numbered in the clockwise direction. So this is the internationally accepted system for numbering of purines and pyrimidine. So purines are numbered in anti-clockwise. Anti-clockwise means, so like this. Clockwise means like this. So that means the number starts in case of pyrimidine in clockwise direction. So that is a 1, 2, 3, 4. So like this. So here it is a 1, 2, 3, 4, 5, 6. That is a clockwise direction. Clockwise direction. But in case of pyrimidine, it is a anti-clockwise. So here the number starts from 1, 2, 3, 4 like this. So that is a anti-clockwise direction. So this numbering for purine and pyrimidine is the accepted internationally. Okay. And coming to the different types of purines which are present in the nucleic acids or nucleotides which are composing the DNA and RNA. So for example, in case of DNA and RNA, what type of purine bases are present in the nucleotides? So here we can see in case of DNA, what type of purines are present. So adenine and guanine. So here I said that, so it consists the heterocyclic atom or heterocyclic rings which are having the 4 nitrogen bases, and at this position, so it is numbered like this. So 1, 2, 3, 4. So this is the 4, 5, 6, and 7, 8, 9. So this is the numbering system for purine. So here at the 6th position, it is having the amine substitution. So that's why that purine is called as the adenine. 6-aminopurine are also called as adenine. And another type of purine moiety which is present in the DNA and RNA nucleic acids are nothing but guanine. So guanine means it is having the amine group at 2nd position. There is a 2-amino, and at 6th position there is a carboxyl group. At the 6th position, so at the 2nd position it is having the amine group, and at the 6th position it is having the carboxyl group. So that's why it is considered, it is named as 2-amino 6-oxypurine. 6-oxypurine also called as guanine. Okay, so these are the 2 types of purines which are present in the nucleic acid structure, that is a DNA and RNA. And coming to the pyrimidines, so pyrimidines, it is having the heterocyclic ring having the 2 nitrogens. Along with 2 nitrogens, if it is having the amine group and carboxyl group at 2nd position, there is a 2nd position is the carboxyl group, and 4th position is the amino group, then it is called as the cytosine. So when it is named like this, there is a clockwise direction. 1, this is the 2, 2nd position, 3rd position, and 4th position, 5th and 6th. So when you see in this, at the 2nd position there is a carboxyl group, and at the 4th position amine group is attached to this pyrimidine ring. So 2-oxy-4-amino pyrimidine, so also called as cytosine. So this is the pyrimidine moiety which is present in the DNA and RNA structure. And the nucleic acids differ with respect to the 2nd pyridine base. So the second pyrimidine base which is present in the nucleic acids is nothing but thymine and uracil. So a second pyrimidine base which is present in the DNA and RNA. So this is the difference in the second pyrimidine base for DNA as well as the RNA. So we know that, so all the purine and pyrimidine bases are present, like adenine, guanine and cytosine is present in the DNA and RNA. But coming to the second pyrimidine base, so this is the first pyrimidine base is nothing but the cytosine which is commonly present in DNA and RNA. But coming to the second pyrimidine base which is present in the DNA and RNA is different. In case of DNA, so what it contains? It contains the thiamine, and in case of RNA it contains the uracil. So coming to the second pyrimidine base, DNA having the different second pyrimidine moiety and RNA has the different second pyrimidine moiety. So DNA contains the thiamine and RNA contains the uracil. Thiamine means it is nothing but a 2,4-deoxy-5-methylpyrimidine. So here we can see at the second position and fourth position there is a carboxyl group, and at the fifth position methyl group is attached to the pyrimidine base. That's why it is called as 2,4-deoxy-5-methylpyrimidine, also called as thymine. Okay, and in case of RNA, the second pyrimidine moiety is nothing but uracil. So it is also named as 2,4-dioxypyrimidine. That means at the second position and fourth position there is a carboxyl group is present in the pyrimidine. So that's why it is called as the 2,4-dioxypyrimidine, also named as the uracil. So that means there are four types of bases are present, and first two are nothing but two are purines and two are pyrimidines. Three types of pyrimidines. In that total 5, 3 are common. So what are the 3? 2 are purines. So that is nothing but adenine and guanine. These two purines are common in both RNA and DNA structure. But and coming to this pyrimidines, one pyrimidine, that is nothing but the cytosine. Cytosine is also called as 2-oxy-4-aminopyrimidine. So it is also common in both the DNA and RNA. So the two pyrimidines and one pyrimidine. Two purine bases and one pyrimidine base. Two purine bases are nothing but adenine and guanine, and one pyrimidine base is nothing but cytosine. So these three are common in DNA and RNA. But the second pyrimidine base, so that is a second pyrimidine base. So that is nothing but the either it is thiamine or uracil. Thiamine or uracil. The second pyrimidine base is maybe thiamine or uracil. So when the thiamine is present, when it is a DNA. So that means if the nucleic acid is a DNA, in that situation the second pyrimidine base is nothing but thiamine. Or the nucleic acid is the RNA. If it is RNA, the second pyrimidine base is nothing but uracil. So based upon that we can characterize whether it is RNA or DNA. Okay, the pyrimidine consisting of the thiamine and uracil differing the structure by the presence of or absence of methyl group. So the difference between the thiamine and uracil is nothing but the presence of methyl group at the fifth position of this pyrimidine. So both otherwise both are same. Okay. That means the major bases, either it is a purine or pyrimidine, present in nucleic acids are nothing but, so in case of purines, the major bases are adenine and guanine. In case of pyrimidine, the major bases present in this RNA and DNA is nothing but, first one is a cytosine and second one is either thymine or it is the uracil. So thymine is present in the DNA and it is uracil is present in the RNA. Okay, so these three are common in both RNA and DNA. So DNA and RNA. But thymine is present in only DNA, uracil is present in the RNA. So these bases, so what are the bases? Purine, in case of purine bases, that is adenine and guanine, and in case of pyrimidine, cytosine, thymine and uracil. These bases are called as the major bases which are present in the nucleic acids. And coming to the some of the minor bases which are found in the nucleic acids are nothing but 5-methyl cytosine, N-acetyl cytosine, N-methyl adenine, N-dimethyl adenine, pseudouracil. So these are the some minor bases which are found in the nucleic acids. So these minor bases help in the recognition of specific enzymes. So by identifying these minor bases, we can identify what type of enzyme it is. Okay, and other biologically important bases are nothing but hypoxanthine, xanthine. So these are the some intermediates in the purine synthesis, and uric acid, that is the end product of purine degradation and present in the free state in the cells. So this uric acid is present in free state in the cells. Okay, and purine bases which are present in the plants are nothing but, so generally the plants contains methylated purines which are having the pharmacological actions like caffeine, theophylline and theobromine. So these are the some purine bases which are generally present in the plant. So like tea and coffee, so that are nothing but caffeine, theophylline and theobromine. So theobromine generally present in the cocoa butter, and caffeine is present in the coffee, theophylline is present in the tea. So these are the structures of hypoxanthine, xanthine and uric acid. So we can see so these are the purine bases. Okay, so that purine bases are present as the free state in our cells. So these are acting, so these are the some intermediates in the purine synthesis, and also the uric acid is the end product of the purine degradation or purine metabolism. So up to now we discussed about the bases, the basic molecule present in the nucleotides. Next, coming to the sugars of nucleic acids. Sugars of nucleic acid. We already know that the nucleotides consisting of, so it is a composition of nitrogenous base, pentose sugar and phosphate. So we already discussed about the nitrogenous bases. There are two types of nitrogen bases, purines and pyrimidines. Okay, and coming to the sugars, there is a pentose sugars. So the sugars which are present in the nucleic acids are nothing but, there are 5 carbon monosaccharides are also called as pentoses. So there are 5 carbon monosaccharides are also called as pentoses. So they are found in the nucleic acid structure. So for example, if it is a RNA, so the RNA contains the D-ribose, but DNA contains the deoxyribose. So the RNA contains D-ribose and RNA contains deoxyribose. So RNA is the ribose one and DNA is the deoxyribose. Deoxyribose sugar is present in the DNA. Ribose and deoxyribose differ in the structure at carbon 2. So that is I can consider this one as the pentose sugar structure. So the ribose and deoxyribose are differ only at carbon 2. So that means the deoxyribose has the one oxygen less than the ribose at carbon 2. So when you observe these structures, so first one is a D-ribose and second one is a D-deoxyribose, and here we can see the numbering of this pentose ring. So that it starts from 1, 2, 3, 4, 5, and at the second carbon, so this is the second carbon. So at this position, the ribose has the two oxygens the ribose has the one oxygen, that is it is attached to the hydrogen and hydroxyl moiety. But in case of deoxyribose, here the oxygen is not present at the carbon 2. So there is a second carbon, so there is no OH group. So the carbon is attached to both the hydrogens. So here it is no oxygen molecule. So that is the difference between the ribose and deoxyribose. So remaining is same in both the ribose and deoxyribose. Okay. And coming to the nomenclature of these nucleotides. So that means naming of the nucleotide, how the nucleotides are named. Okay. So it consisting of the pentose sugar and there is a nitrogenous base, and these two are combined to form the nucleoside. Nucleotide means so nucleotide means it consisting of the nitrogenous base, pentose sugar and phosphate. Nucleotide, but nucleoside means so without the phosphate group, that is the absence of phosphate group. That means it consisting of only nitrogenous base and pentose sugar. So this one is called as the nucleoside. Pentose sugar plus nitrogenous base are combined to called as the nucleoside. So if the sugar is ribose, then they are called as ribonucleosides. So here when you see in this, so the combination of base and pentose sugar, in that sugar, if the sugar is ribose, then this nucleoside is called as a ribonucleoside. Okay, so like adenosine, guanosine, cytosine and uridine are the ribonucleosides. Adenosine, guanosine, cytidine and uridine. So these are the ribonucleosides of adenosine, of adenine, guanine, cytosine and uracil. So these are the adenine, guanine, cytidine and uracil is the nucleotides, but nucleosides of these corresponding nucleotides are nothing but, so the adenosine is the nucleoside of adenine, guanosine is the nucleoside of guanine nucleotide, cytidine is the nucleoside of cytosine, and uridine is the nucleoside of uracil. Okay, so these are the nucleosides of corresponding nucleotides. So that is a ribonucleosides. These adenosine, guanosine, cytidine and uridine consisting of ribose pentose sugar in the that is attached to the corresponding nucleotides. And if the sugar is deoxyribose, the deoxyribonucleosides are produced. So the term mononucleotide is used when the single phosphate moiety is attached to the nucleoside. Mononucleotide means only one one phosphate group is or single phosphate moiety is attached to the nucleoside, then it is called as the mononucleotide. So this is the mononucleotide. So this term is nothing but, so when you apply this term to the nucleoside, to these nucleosides, when only one or single phosphate moiety is attached, in that situation it is, that moiety is called as the mononucleotide. Thus adenosine monophosphate, so that's why the name is given to the AMP, that is a adenosine is the nucleoside and one phosphate is attached, then it is called as the adenosine monophosphate. It consisting of adenine, ribose and phosphate, only one phosphate, that's why it is called as the monophosphate. Adenosine is the nucleoside and it is attached to the one phosphate, that's why it is called as the adenosine monophosphate. This is the one of the nucleotide. So one of the nucleotide, that is a adenosine monophosphate is one of the nucleotide. So that is also denoted by AMP. It is denoted by AMP. So here we can see the different types of nucleosides and nucleotides which are present in different. So this table indicates the different types of bases, nucleosides are present in the RNA and DNA. So that is nothing but the nucleic acids. Okay. So and their abbreviations are given here. Okay. Sorry, present in the nucleotides. So what is the corresponding base and nucleoside are present in the nucleotides, so that are given in this table. So for example, in case of AMP, that is a adenosine monophosphate. Adenosine monophosphate, the base is nothing but adenine, that is a nitrogenous base, that is a adenine, and ribonucleoside, that is a adenosine. Ribonucleoside is a adenosine, that means it is attached to the ribose sugar, that is attached to the ribose sugar, that's why it is called as the adenosine, or it is also called as nucleoside. So when it is ribose pentose sugar is present in the nucleoside, then it is called as a ribonucleosides. But when the deoxyribose is present, then the nucleoside is called as deoxyribonucleoside. Okay. So in case of DAMP, then that is adenine is a base and deoxyribose is the sugar. Then that's why it is denoted as DAMP. It is a deoxyadenosine monophosphate or deoxyadenylate. Okay, so that is in this way we can give the names and some abbreviations. And coming to the synthesis of purines, and these purines are synthesized by the most of the tissues, and the major site of purine synthesis is nothing but the liver. And that liver, the cells of the liver also, the subcellular site, that is in that cells of the liver, the major part involved in the purine synthesis is nothing but the cytoplasm. That means the purine synthesis is majorly occurred in the cytoplasm of the liver cell. So there are different types of pathways for synthesizing the purines. First one is a de novo synthesis and second one is a Salvage pathway. There are two major pathways. Two pathways are present in the purine synthesis. First one is a de novo synthesis. Second one is a Salvage pathway. In that two, de novo synthesis is the major pathway. So that means most of the purines are produced by this de novo synthesis process. And Salvage pathway is the minor pathway. So few amount of purines are produced by this Salvage pathway. The synthesis of purine nucleotides from various small molecules derived as the intermediates of many metabolic pathways in the body. So that means the purine nucleotides are produced from various small molecules which are derived from the intermediates of many metabolic pathways. So there are some metabolic pathways happened in our biological system. Through that metabolic pathways, some intermediates are produced. from these intermediates, these starting materials of purine nucleotides are obtained. So the synthesis of purine nucleotides is nucleotides from various small molecules derived as the intermediates in many metabolic pathways of our body. Okay. Next, we'll see how the de novo synthesis is producing the purines. So de novo synthesis, de novo means generally the meaning for de novo is nothing but new synthesis or new synthetic method of the purines. So purine, that means new purine molecule is synthesized. That means there is no existence of the any this nucleoside or what is a nitrogen base is exist. So the nitrogen base also newly synthesized. That's why the name is given to this pathway as de novo synthesis. So in Salvage pathway, what will happen? On the existed nitrogen base, the remaining structure is developed. But here, the nitrogen base also produced as the new compound, or the new nitrogen base also produced on the sugar moiety. That's why this name, there is a de novo synthesis is given. So that means there is no utilization of the nitrogen base for the synthesis of these nucleotides. Okay, so it will create the new nitrogen base on the 5, that is a ribose 5 phosphate. The furin ring or the base ring is built on the ribose 5 phosphate. So generally the base present in this purines is nothing but the adenine and guanine. Okay, so this type of purine rings are built on the ribose 5 phosphate. So here when you observe this purine ring here, so when you observe the purine ring, so there are different atoms are present in the purine ring. That means 4 nitrogens are present and remaining carbons are present in the 2 aromatic rings. So each and every atom is donated or contributed by the different type of amino acids and some other molecules. So for example, when you see in the nitrogen, so that is indicated by nitrogen 1. When you number these rings, starting from 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 atoms present in this purine ring. So each and every atom is donated or contributed by a particular moiety. So the first nitrogen is donated by the aspartate, and second carbon is donated by the N10 formyl tetrahydrofolate, and third nitrogen is produced from the glutamine, and fourth is produced from the sugar moiety, and fifth also produced from the sugar, and sixth is produced from the carbon dioxide, and this glycine will produce the seventh nitrogen, and N10 formyl tetrahydrofolate is again producing the contributing the 8th carbon, and glutamine contributing the again 9th carbon. So these are the some compounds or substances can contribute the different atoms which are present in the purine ring. Okay, so let us see how they are contributing in a sequence manner. The parent purine nucleotides first synthesize the inosine monophosphate. So inosine monophosphate is a parent purine nucleotide. Parent purine nucleotide is nothing but inosine monophosphate. So by this inosine monophosphate, so we can able to produce the other nucleotides, that is a purine nucleotides, nothing but the AMP and GMP, like adenosine monophosphate, guanosine monophosphate. So the AMP and GMP are produced from the utilization or from the inosine monophosphate. So when you see in the inosine monophosphate, it is nothing but the composition of hypoxanthine, ribose and phosphate. So hypoxanthine is the base, nitrogenous base, and ribose is the sugar and phosphate. So this is the composition of inosine monophosphate. So by utilizing this parent purine nucleotide, we can synthesize the other nucleotides like AMP, GMP. So first we'll see how the inosine monophosphate is produced or synthesized. And these are the sequence of steps which are involved in the biosynthesis of inosine monophosphate. So there are 10 steps are present. Each and every step will contribute. That means each and every step will contribute one or two atoms that are present in the purine ring. So in first step, there is a addition of ninth nitrogen. This step is called as a rate limiting step, and so thereby it can produce the pyrophosphate ribosyl adenylate. And second one is a glycine. So glycine is the amino acid which can produce the carbon 4, 5 and also nitrogen 7. Okay, so that is a, so glycine is the, I think it is a pentose sugar moiety which can produce the carbon 4, 5 and nitrogen 7. So that is a adenylate guano adenylate ribosyl. Okay, it produces that moiety. So like that, for each and every step there is a one substance is involved to contribute the corresponding atom which is present in the purine ring. So here we can see the series of steps involved in the purine synthesis. Purine synthesis. And in the first step, ribose 5-phosphate. So it is obtained from the HMG shunt of carbohydrate metabolism. So this ribose 5-phosphate reacts with the ATP to form phosphoribosyl pyrophosphate. So here it is a ribose 5-phosphate. It can react with the ATP and can able to form the, what is that? Phosphoribosyl pyrophosphate, PRPP. So this step is nothing but the formation of PRPP. Formation of the PRPP. This one is a rate limiting step. And in the second step, there is a formation of 5-phosphoribosyl amine. So that is a, so there is a glutamine transferase, one of the enzyme, so that can add amide nitrogen to the phosphoribosyl pyrophosphate. Glutamine transferase, this is the enzyme. What it do? It can add amide nitrogen to the phosphoribosyl pyrophosphate to replace the pyrophosphate, group. Thereby it can produce the 5-phosphoribosyl amine. 5-phosphoribosyl amine. So this is the 5-phosphoribosyl amine. So 5-phospho. So this moiety, 5-phosphoribosyl amine. So what it will do in this pyrophosphate group is removed by the addition of amide nitrogen. So thereby the molecule is converted to 5-phosphoribosyl amine. So in this step there is addition of nitrogen group is attached. There is a nitrogen group is attached to this in this step. So this one is considered as the committed step. And next one is the phosphoribosyl amine. Phosphoribosyl amine is reacted with the glycine in the presence of ATP. So here this step. So the phosphoribosyl amine will react with the glycine in presence of ATP to form the glycinamide ribosyl 5-phosphate. Glycinamide ribosyl 5-phosphate. So during this process, this is a glycinamide ribotide is formed. That is nothing but the addition of carbon 4, carbon 5 and nitrogen 7. So the 4th carbon, 5th carbon and 7th carbon is added in this step. So that is nothing but the conversion of phosphoribosyl amine to glycinamide ribosyl 5-phosphate. And in the next step, to this formed glycinamide ribosyl 5-phosphate, So there is a addition of pharmal group. So the addition of pharmal group is occurred by the N10 pharmal tetrafolate. N10 pharmal tetrafolates. So this is the substance which can donate the pharmal group, and the product formed by this addition or donation is nothing but the pharmyl glycinamide ribosyl 5-phosphate. So this is the last compound. So in this image, so that is nothing but pharmyl glycinamide ribosyl 5-phosphate. And the next step, there is a transfer of second amino group. Second, that is amino group. So that is occurred from the glutamine. So to the pharmyl glycinamide ribosyl 5-phosphate, there is a transfer of amino group. So from the glutamine, so that will form the pharmyl glycinamide ribosyl 5-phosphate. So pharmyl glycinamide sorry, not a glycinamide. So it is a pharmyl glycinamidine ribosyl 5-phosphate. Pharmyl glycinamidine ribosyl 5-phosphate. Then the imidazole ring is closed by the consuming of ATP. There is a ATP dependent reaction. So thereby it can release, it can produce the 5-amino imidazole ribosyl 5-phosphate. So this is the closing structure. So the imidazole ring of the purine is closed. So this process is the ATP dependent reaction. That means it utilizes the ATP for the closing of purine ring of the imidazole ring of the purine. So thereby it forms the 5-amino imidazole ribosyl 5-phosphate. And in the next step, the carbon dioxide. So by the contribution of carbon dioxide, it yields amino imidazole carboxylate ribosyl 5-phosphate. So that means the carbon is donated from the carbon dioxide. So thereby the previous molecule, that is nothing but 5-amino imidazole ribosyl 5-phosphate, is converted to amino imidazole carboxylate ribosyl 5-phosphate. Then in the next step, the aspartate will condense with the product. So that is the aspartate will condense with the amino imidazole carboxylate ribosyl 5-phosphate. So with this product, so there is a amino imidazole carboxylate ribosyl 5-phosphate. The aspartate is condensed to form the amino imidazole 4-succinyl carboxamide ribosyl 5-phosphate. So there is the this product. There is the last product of in this image. So there is a amino imidazole 4-succinyl carboxamide ribosyl 5-phosphate. So this is the formation of. So that means in this reaction there is a addition of another nitrogen. There is a nitrogen at the first position is done in this reaction. So the nitrogen at the first position is contributed from the aspartate. When the aspartate is reacted with the amino imidazole carboxylate ribosyl 5-phosphate, so during the condensation reaction or with the condensation process. In the next step, the adenosuccinate lyase. So that is an enzyme that cleaves the 5-amino imidazole 4-succinyl carbamide ribosyl 5-phosphate. So that is a previously formed compound in the 8th step. That is a 5-amino imidazole 4-succinyl carboxamide ribosyl 5-phosphate. So this molecule is cleaved by the enzyme, that is adenosuccinate lyase. So this will cleave the fumarate group. The fumarate group which is present in this 5-amino imidazole 4-succinyl carboxamide ribosyl 5-phosphate. Thereby it is converted to iminoimidazole 4-carboxamide ribosyl 5-phosphate. That is a, so in this process there is a removal of fumarate molecule of the aspartate. Okay, so only the imino group is present, the imino group of aspartate is retained, the fumarate molecule is removed in this process or in this step. And next in the next step, there is a donation of 1 carbon moiety, so that is obtained from the N-pharmyl tetrahydrofolate. So thereby it can produce the pharma-amino-imidazole 4-carboxamide ribosyl 5-phosphate. Pharma-amino-imidazole 4-carboxamide ribosyl 5-phosphate. So that means the second carbon is added in this reaction from the N10-pharmyl tetrahydrofolate. Okay, so with this reaction all the carbons and nitrogens of this purine moiety is attached, and there is a one thing is required, that is a completion of the ring, that is a closure of the ring. So that is done at the, so that is happened in the final step. There is a final reaction of the catalyze, so that is catalyzed by this cyclohydrolase enzyme. Cyclohydrolase enzyme, so that will catalyze the reaction of cyclization. Thereby the ring is closed and it can able to form the inosine monophosphate. So the cyclohydrolase is the enzyme that can convert the 5-pharma-amino-imidazole 4-carboxyamide ribosyl 5-phosphate into inosine monophosphate by ring closure reaction, by ring or cyclization reaction. So this is the steps involved in the biosynthesis of the parent nucleotide, that is nothing but the inosine monophosphate. So from this inosine monophosphate we can produce the other nucleotides like AMP and GMP. And here the synthesis of AMP and GMP from the IMP is generally happened. So the aspartate condenses with the IMP in the presence of GTP to produce the adenosyl succinate, so which on cleavage forms the AMP. So that means this is the single step reaction, that is aspartates condense with the IMP. So first we will already seen that or discussed about the biosynthesis of inosine monophosphate. After formation of the inosine monophosphate, so it will act as a precursor for the formation of AMP and GMP. And the formation of AMP, so the inosine monophosphate is condensed with the aspartate in the presence of GTP, so to produce the adenyl succinate. So when the inosine monophosphate condenses with the ATP in the presence of GTP, it produces the adenylosuccinate, and this adenylosuccinate cleaved by the enzyme, thereby it forms the AMP. And in case of synthesis of GMP, so for the synthesis of GMP, the IMP, that is inosine monophosphate, undergoes NADP dependent dehydrogenation, so to produce the xanthanoase monophosphate, and the xanthanoase monophosphate will receive the amide nitrogen from the glutamine to produce the GMP, that is a guanosine monophosphate. So the guanosine monophosphate is produced by the NAD plus dependent dehydrogenation process of inosine monophosphate to produce the xanthosine monophosphate. And after formation of the xanthosine monophosphate, this xanthosine monophosphate will receive amide nitrogen from the glutamine, thereby it will be converted to guanosine monophosphate. So here we can see the reactions that can be produced adenosine monophosphate and guanosine monophosphate from the inosine monophosphate. So the first inosine monophosphate is converted into adenylosuccinate. So this will be done by the condensation of inosine monophosphate with the adenosine. So adenosine by the enzyme activity like adenosine succinate synthase. So inosine monophosphate will condense with the adenylate with the action of adenylosuccinate synthase. Thereby it will be converted to adenylosuccinate. Then it will be cleaved. The adenylosuccinate is cleaved by the catalytic activity of adenylosuccinase. So thereby it will be converted into adenosine monophosphate. There is a cleavage will takes place here by the enzyme, that is adenylosuccinase. So then it will be converted into adenosine monophosphate. And next, so in case of GMP formation from the inosine monophosphate, so first there is a conversion of inosine monophosphate to xanthosine monophosphate by the NAD plus dependent dehydrogenase. So for that, dehydrogenase. So for this, there is a dehydrogenase reaction is involved in this reaction. So that will undergoes dehydrogenation process of this inosine monophosphate to xanthosine monophosphate. So after formation of the xanthosine monophosphate, this xanthosine monophosphate will receive the amine nitrogen. So it will, this xanthosine monophosphate, that is the XMP, will receive the amine nitrogen from the glutamine. So this glutamine will contribute the amine nitrogen. So thereby it will be converted to xanosine monophosphate. So these are the steps involved in the biosynthesis of adenosine monophosphate and guanosine monophosphate from the inosine monophosphate. So the inosine monophosphate is a parent nucleotide. It acts as the precursor for the synthesis of adenosine monophosphate and guanosine monophosphate. And coming to the inhibitors of this purine synthesis. So some of the inhibitors like sulfonamides are the structural analogs of the para-aminobenzoic acid. So these sulfur drugs can be used for the inhibitors for inhibiting this synthesis of these purines in the microorganisms. So this indirectly reduces the synthesis of purines and the nucleic acids in microorganisms. So these sulfonamides that doesn't show any influence on the humans, since there is no folic acid is synthesized or supplied through the diet for humans. And the structural analog of folic acid, that is a methotrexate, is widely used as the agent to control the cancer in humans. So this methotrexate will inhibit the synthesis of purine nucleotides and other nucleic acids in the humans. And these inhibitors are also affect the proliferation of the normal growing cells also. In some situations, this folic acid analog, that is a methotrexate, may affect the proliferation or growth of the normally growing cells. So this is the this is about the biosynthesis of purine nucleotides and their inhibition. Okay. So in next class we'll discuss about the what are the biosynthesis of pyrimidine nucleotides. So in next class we'll see about the biosynthesis of pyrimidine nucleotides. Thank you.